all the proteins that are involved in going from reception to the cellular output response are part of what's called signal transduction. All right. When I was showing you this signal transduction pathway here, I showed you this receptor right here activating the first protein, and then the first relay protein activates the second protein, and that one activates the third relay protein. Then eventually we get to activating the cellular response. Maybe I should talk about, well, what does it mean when we say a protein is activated? How do you activate a protein? Actually, before I get into that, maybe I'll just point out something here. In this particular signal transduction pathway, I'm showing just 3 relay proteins between the receptor and the cellular response protein. But that's not always, I don't mean to be saying it's always going to be 3. In many signal transduction pathways, there might be many, many more relay proteins between the receptor and the cellular response protein. Okay, so I was going to talk about, well, what do we mean when we say a protein is activated? Well, this is something that we actually talked about in a previous chapter, I think on the chapter on enzymes and metabolism. Oh yeah, so the whole thing's a signal transduction pathway. And here we go. One way in which an enzyme can be activated is by these things called allosteric activators. And the idea here was this, that certain enzymes in their natural normal shape have a closed binding site. Here's the substrate that this enzyme is supposed to be working with, but the enzyme's binding site just isn't open enough for the substrate to fit in, so the enzyme has no activity in its normal natural state. But there are molecules called allosteric activators that come along, and by definition, allosteric activators don't bind directly to the active site. They plug in at some other location. You can think of like the back of the enzyme. And when they bind, they end up pulling on the amino acids of the enzyme, and that opens up its active site, like that. And so now we say the enzyme has been activated. Its active site opens up, and it can carry out its particular activity. So when we saw that signal transduction pathway, we saw enzymes getting activated. In some case, that means allosteric activators were generated inside the cell, and that's how the enzymes became activated. Here's a point that I think I mentioned when I covered this a couple of weeks ago. The allosteric activator is not covalently bonded to the enzyme. There might be hydrogen bonds or perhaps ionic interactions keeping it in here, but no sort of permanent covalent bond. This is not considered a permanent part of the enzyme. Another way that an enzyme can be activated is through phosphorylation. Here's an example of that. Here's yet another enzyme where, don't do that. This is supposed to be another enzyme where the active site is closed in the enzyme's normal, natural, folded position. But if we want to get this enzyme activated, what can happen is a phosphate group can be added to some of its amino acids. And this is a covalent bonding. When a phosphate group becomes attached to amino acids on an enzyme, there is actually a new covalent bond between the two. It's a more permanent linking than we saw with the allosteric activators. But the result is the same. This new functional group on this particular amino acid causes the different interactions between it and the rest of the amino acids, and so the shape of the enzyme can change, and that can open up the active site, and now the enzyme is going to be able to carry out its particular reaction. So in those signal transduction pathways I was showing you, when I say the receptor activates this relay protein, and this relay protein activates another relay protein, Sometimes what we're talking about is that the enzymes are becoming activated by getting phosphorylated, and other times what I'm talking about is the enzymes are getting activated because of allosteric activators. Alrighty, well, questions up to this point? Alright, well, let's get into even more depth about this. A lot of research has been done. I'm going to come back to this part about the kinases here in a few minutes. I want to skip down to this part right here. A lot of research has been done into signal transduction pathways, and they studied a lot of different signal transduction pathways that involve many different signal molecules and many different receptors, many different cellular output responses. And what they found is that there are three major families of signal transduction pathways based on the types of receptors that start the signal transduction pathway. These three types of receptors are called tyrosine kinase receptors, G protein link receptors, and ion channel receptors. So let's start with the first one and see what its particular signal transduction pathway looks like, and then we'll focus in on G protein receptors when we finish that. All right, let's see. There they are. Tyrosine kinase receptors. All right, so here's a cell, and here's its membrane, and we'll say that's the inside of the cell toward the bottom, and that's the outside of the cell toward the top. And we will give it one of these tyrosine kinase receptors. There it is right there. The little T's at the bottom of the receptor represent the amino acid tyrosine. Tyrosine, remember that there are 20 different types of amino acids, and tyrosine is one of the 20 types of amino acids. So all tyrosine kinase receptors have some tyrosine amino acids as part of their structure, which is not to say that any protein that has tyrosines is a tyrosine kinase receptor. Lots of proteins have tyrosines. That's actually a fairly common amino acid, but I'm just saying all tyrosine kinase receptors have to have tyrosines as part of their structure. All right, so like all receptors that we've talked about, it has an extracellular domain that sticks out of the cell, and a hydrophobic transmembrane domain, and an intracellular domain sticking down into the cytoplasm. When the signal molecule comes along, that's going to be detected by a tyrosine kinase receptor, comes along like this, sticks to the binding side of the receptor. Here's where things get interesting. In and of itself, the signal molecule binding to the receptor is not enough for the intracellular part of the receptor to start the signal transduction pathway. If this is all there was, just nothing would happen. No signal would be generated inside the cell. What has to happen is this. Another tyrosine kinase receptor that's also bound the same signal molecule will come over, and the two will now click together like this. And this is called forming a dimer, or you say that the tyrosine kinase receptors have dimerized. And the word di means two, so this just means forming a pair. And when that happens, when the two tyrosine kinase receptors that have bound their ligand, their signal molecule, have dimerized, then that effectively forms the active site of an enzyme. So when there was just one, before they had dimerized, there was no enzymatic activity of the intracellular domain because there wasn't really an active site there. But when they click together, then an active site is formed. And so now working as a dimer, working as a pair, these two receptors are now going to start functioning as an enzyme. They're going to be able to carry out a chemical reaction. And the chemical reaction that they're going to be able to carry out now is something called tyrosine kinase enzymatic activity. What the heck is that? Well, flipping backwards to to this one. Here a moment ago I was telling you that certain enzymes get activated by getting phosphorylated, or phosphorylation, which is when a phosphate group is attached to one of their amino acids. Well, any enzyme that attaches phosphate groups to the amino acids of a protein is called a kinase enzyme. Kinase enzymes take phosphates and stick them onto amino acids of proteins. Kinase enzymes are phosphorylating enzymes, and that's a phosphorylate. And where do they get the phosphates from? Well, ATP. Remember that ATP molecules have a phosphate. So ATPs have 3 phosphate functional groups. And so ATP is the source, and here we'll see it. So this, when they dimerize, this region right here is now a functional tyrosine kinase that's able to add phosphates to tyrosines. And what's actually going to do is add them to its own tyrosines. So it will grab an ATP, rip off one of the phosphates, and add it to its own tyrosine. Of course, you get an adenosine diphosphate left over, but we're not going to use that for anything. It just drifts off. There's ATP. And the tyrosine kinase enzyme activity here at the bottom of the dimer keeps going. It keeps phosphorylating all its own tyrosines. So it'll continue to grab ATPs until they all have phosphate groups on them. Like that. Now, maybe I should stop here for a second and point something out. So the term kinase means any enzyme that's able to add phosphate groups to amino acids. And so this is certainly acting as a kinase. The dimer is acting as a kinase. And in particular, it's a tyrosine kinase because that means kinases that add phosphates to tyrosine amino acids. But notice that this particular tyrosine kinase is phosphorylating its own tyrosines, and that is a little unusual. Your cells have lots of kinase enzymes in them, but almost always those kinase enzymes phosphorylate other proteins, not themselves. But for some reason, this one autophosphorylates. It's a tyrosine kinase that phosphorylates its own tyrosines. I don't know why that is, but it is. But just as I said, bear in mind that most tyrosine kinases phosphorylate other proteins' tyrosines, not their own. Okay, so now the dimer has phosphorylated itself. Here we go. And remember that after the receptor, the next step is to activate relay protein number 1, the first relay protein. And these phosphorylated tyrosines are the activators of the first relay protein. In other words, relay protein number 1 is going to come along, contact those phosphates, and that's going to cause a change in the shape of relay protein number 1. So it now becomes an activated relay protein. And so now it's going to go off and activate relay protein number 2, which will activate relay protein number 3, which will activate relay protein number 4, until we finally get to the cellular response protein. And I should point out that's also unusual. Notice that relay protein number 1 in this case didn't have to itself get phosphorylated. It just merely had to contact the phosphates that were stuck on the tyrosines of the receptor, and that was enough to activate it. And that is unusual because most proteins that are activated by phosphate groups have to themselves get phosphorylated, not just brush up against some other protein's phosphates. So I almost hate to use tyrosine kinases as an introduction to all this because they're unusual. They are one of the few kinases that autophosphorylates, and their phosphate groups then activate another protein, not the enzyme itself. Okay, well, one quick closing thought here. Let's go right here. So then that first relay protein moves away from the tyrosine kinase receptor. It'll activate the next relay protein, which activates the next relay protein. in what way is this first relay protein activating the next relay protein? Well, this first one can be a kinase. It might be a tyrosine kinase that phosphorylates the next one's tyrosines, like this. It grabs an ATP to get some phosphate groups, adds them to the tyrosine amino acid on relay protein number 2, and that activates relay protein number 2. So we would say that this relay protein right here is a tyrosine kinase. And give me one more minute. There's another type of kinase called a serine-threonine kinase. This was a serine-threonine kinase if it adds phosphates to serines and threonine amino acids on a protein to activate that next protein. So those are the two major types of kinases. You have tyrosine kinases and serine-threonine kinases, depending on which amino acids get phosphorylated. Okay, you guys are good. Thank you, and I will see you in the laboratory.